Welcome to the journey where we explore DNA and RNA replication. So, what is DNA? DNA or deoxyribonucleic acid is a self-replicating polymer which is the carrier of genetic information. Now, I'll be discussing structure of DNA. DNA is a polymer made up of nucleotides. First, we'll see what's a nucleotide. A nucleotide is a triad of a nitrogenous base, pentose sugar, and a phosphate group. There are four types of nitrogenous base, that is adenine, guanine, thymine, and cytosine. Accordingly, there are four types of nucleotides: thymidine, cytidine, adenosine, and guanosine. Thymidine and adenosine are pyrimidines, whereas cytidine and guanosine are purines. Now, you must be familiar with the double helix model of DNA. If you untwist it, you get the stepladder model. We will first get familiar with the stepladder model, and that will be similar to getting familiar with the double helix model. Now, you might wonder that how two nucleotides in the same strand are joined together. The carbon 5 of one of the nucleotides is joined with the carbon 3 of the other by a phosphodiester bond, and the sugar phosphate group forms the backbone of the nucleotide. Directionality and antiparallel bases. Directionality means that if one of the strand goes from 3' to 5' end, the other has to go from 5' to 3' end. Now, base pairing. Base pairing means that an adenine molecule has to bond with a thymine, and a cytosine molecule has to bond with a guanine. Now, what is the advantage of base pairing? The advantage of base pairing that it gives a structural advantage to the nucleotide and to the DNA. Now, before we move on, I'd like to emphasize some differences between DNA and RNA. Now, the first and foremost difference is that DNA is double-stranded, whereas the RNA is single-stranded. Now, DNA consists of a thymine, whereas RNA consists of uracil, and at the second carbon of DNA there's an H group, whereas at the second carbon of RNA there's an OH group. So, DNA replication. Now, where and when does this take place? Now, the first answer is kind of obvious, that it takes place in the nucleus. But when does it take place? It takes place during the interphase of the cell cycle, just before the cell division. Now, the major enzymes in DNA replication are DNA polymerase, helicase, primase, and ligase. Don't worry about the name, you'll get a hang of them later. Now, helicase, it is responsible for opening up of DNA to allow replication of it. It is the unzipping enzyme. Primase, it catalyzes the synthesis of a short RNA segment called primer. Complementary to a ssDNA template, it tells the DNA polymerase where exactly to start synthesis. DNA polymerase is the builder enzyme. It creates two complementary DNA duplexes from a single original DNA duplex. Ligase. DNA ligase is an enzyme which can connect two strands of DNA together by forming a bond between the phosphate group of one strand and the deoxyribose group of another. It is used in cells to join together the Okazaki fragments which are formed on the lagging strand during DNA replication. Now, you'd ask me, what are these Okazaki fragments and lagging strand? Well, I'll come to it later. Before proceeding to the actual process of DNA replication, I just want to emphasize that DNA polymerase can only act from 3' end to 5' end. it cannot synthesize a new strand from 5' end to 3' end. In the process of DNA replication, the DNA helicase comes into play and it unzips the two strands. Now, there are proteins such as the SSB proteins that prevent the two strands from getting together again, and there are enzymes such as topoisomerase which prevent the DNA to supercoil. Now come the DNA primase. The DNA primase catalyzes the synthesis of a primer RNA which tell the DNA polymerase where exactly to start the synthesis. I just want to emphasize that this strand runs from 3' end to 5' end. So DNA polymerase will have no issues while synthesizing its complementary strand. But what about this strand? It will have to work backwards and work in segments. That's why this strand is called the leading strand, whereas this one is called the lagging strand. As you can see that in the leading strand there's a complete DNA molecule, but in the lagging strand, is it so? No. The molecules are in fragments and these fragments are called the Okazaki fragments. Now, do you remember that I told that an enzyme fixes this? Yes, you are right. It's the ligase which glues the Okazaki fragments together. Here we have completed with DNA replication. Now, I just want to emphasize that DNA replication is semi-conservative. That means one of the strands is from the original DNA and the other strand is newly synthesized. Here you can pause the video and summarize whatever we have learned. Now let's get started with RNA synthesis. Now again the question comes, where and when? Well, where is kind of obvious again that it happens in the nucleus, but when? It occurs during protein synthesis. Now, it's very important. Protein synthesis has two steps, transcription and translation. Transcription is the first step in gene expression or protein synthesis. It involves copying of genes in DNA sequence to make an RNA molecule, also known as mRNA or messenger RNA. Now, RNA polymerase is the most important enzyme in RNA replication. It copies the sequence of DNA to an RNA during the process of transcription. Now, I want you to see that the strand which goes from 5' end to 3' end is called the coding strand, whereas the one which goes from 3' to 5' is called the template strand. The RNA polymerase gets itself attached to the 3' to 5' end one, or you can also say that it gets attached to the template strand and starts synthesizing a RNA which codes for exact opposite codons. I also want to emphasize that in the RNA, thymine is attached to adenine, but here adenine is attached to uracil. This means that thymine is absent in RNA whereas uracil is absent in DNA. Uracil is exclusive to RNA whereas thymine is exclusive to DNA. This way, the RNA polymerase completes the RNA synthesis. Now, do you remember that the DNA polymerase required a primer to know where exactly to start the synthesis? So, does that mean that RNA polymerase doesn't require such an initiating factor? No, it's not the case. The initiating factor is the AUG codon, which codes for the protein methionine. There's also a terminating factor that are codons UAA, UAG, and UGA. Here we have completed with RNA replication. Now, I just want you to pause the video and have a quick look at this diagram so that you'll remember it for a longer time. Well, I haven't mentioned about protein synthesis, but I encourage you to explore with further reading. Thank you.